So we're going to start discussing first the simpler of the two proteins, the two oxygen binding proteins, and that's myoglobin. And so the ligand binding site for myoglobin is very similar to that in hemoglobin. So this gives us, so we're not changing much by discussing myoglobin instead of hemoglobin. So on the slide here, on the right-hand side, is a crystal structure of myoglobin. And so there are a lot of different these cylinders within the structure here are helices. So these represent helices. And so you can see that sort of the fold that's important in the secondary structure details for myoglobin within this crystal structure. And so on the bottom left-hand corner is the N-terminus or the amino terminus, and then the upper left-hand corner is the carboxy terminus. So it folds around, and then this is the structure. So what's really important, though, what's really critical for O2 binding is this molecule in the center, which is called heme. And we'll actually use, oh yeah, why not? We'll use red for that because it's heme. Okay, and so this is known as a cofactor. So this is a molecule that is not an amino acid. It's not part of the polypeptide chain, but binds to the protein. And in this case, it's really critical for the function because without the heme cofactor, this protein could not bind O2. It's super important. And so the structure of the heme cofactor is actually kind of complicated and kind of interesting. So the center of it are 4 nitrogens. Which have 4 role rings. And then these are connected to each other. Not only that, but this is a conjugated system where 2 of these nitrogens are deprotonated and 2 of these nitrogens are protonated. So if we start here and we start conjugating, so this is protonated. What did I mess up on? That's it. Okay. And then finally, there are groups on the outside here. So there's a propionate group. Methyl groups. And vinyl groups. And so that is the structure of heme, or rather protoporphyrin 9. And what makes it heme is when these 2 nitrogens are deprotonated, and in the center is iron. And these form, the nitrogens form what are called coordination bonds to iron, and this is heme. Okay, and so this is again, this is critical for binding of the oxygen because the oxygen doesn't bind to the polypeptide backbone. It doesn't bind to any of the side chains. What it actually binds to is the iron of the heme, and the heme cofactor is really important and allowing for the stable binding of oxygen, and also for the function of these O2 carrying proteins, such as myoglobin and hemoglobin.